This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
Good. Well, welcome. It's great to see you guys. I want to tell you a little bit about what's going on in my laboratory. And in my lab, we're really interested in trying to understand how cells behave normally when they're building our tissues, and then how they misbehave in diseases like cancer and Alzheimer's disease. So you're not really aware of it, but every single day, every one of the trillions of cells in your body is making an active decision whether to live or die. And you're probably thinking, live, I want them to live. But in fact, it's important that some cells die. So if cells survive that aren't supposed to, we can end up with diseases like cancer or autoimmune disease. And so organisms have evolved cell suicide programs. So they have the capacity to commit suicide for the greater good of the organism. And the most famous one is called apoptosis. But of course, if too many cells die, this causes problems too. And this can lead to degenerative diseases, the common ones that you hear about, Alzheimer's, Parkinson's, etc. And it can also lead to the dire consequences that follow a heart attack or a stroke. So of course, cell survival mechanisms are really important too. And I want to tell you about one that we've been studying in my lab, and that we've discovered that cells can actually recover from the brink of apoptotic cell death. And what we think, I'll show you some examples of this, and why we think this might be important. Okay, so first I have to tell you about apoptosis. This is this program that cells have where they can commit suicide if they are in response to various stimuli. So here's a schematic of a cell with its nucleus, and it was described, this process of apoptosis was described and named many years ago, decades ago. And it was initially described as a series of changes in the visible shape and appearance of the cell. So the cell shrinks, the membrane starts making these little bubbles and that are called blebs, and then the nucleus condenses, and things start to break apart, and then the cell breaks apart into these so-called apoptotic bodies. And these bodies are then consumed by the professional sort of garbage men in the cellular world, which are cells like macrophages and other cells that eat up the remains of the dying cell. Now, we now know, after many years of study by many people, that these changes in the appearance of the cell as it's committing suicide are really driven by a series of biochemical changes inside of the cell. And most importantly, no matter how cell death is initiated, and in an adult organism, you can think of it being initiated by certain types of injury, like a response to radiation, or toxic chemicals that are in the environment, or oxygen-free radicals, and those kinds of stimuli can cause the cell to commit suicide by this mechanism of apoptosis. And we know that the key event inside of the cell is the activation of this particular class of enzymes called executioner caspases, right? They're the executioners, and once these enzymes are activated, they run around the cell and start chopping things up, literally. They're proteolytic enzymes, and they cut other proteins up into pieces. And this leads to those visible changes in the cell that we saw, where the DNA starts to break down, where the membrane starts to bleb out, and the cell starts breaking up into pieces. So in the field of apoptosis research, it's widely believed that once these caspase enzymes are activated and initiate these morphological changes, that this is an irreversible step and that there's no going back. But a former postdoctoral fellow in my laboratory really thought that had been an assumption and that had not been adequately tested. So he did a pretty simple experiment. He wanted to know, is it really true that once cells undergo these changes in visible shape, once caspase are activated, is it really impossible for these cells to recover? So he did a simple experiment, as shown here, and he got a really striking result. Here we're looking at a cell in the microscope. This is a primary cell from a mouse taken straight out of the animal. It's labeled with two fluorescent dyes that you see here, one in the nucleus that's bound to DNA, and the other one, the red one here, is labeling the mitochondria, right? Those are the little energy factories in the cell, and they're incredibly important during the process of cell death. During apoptosis, the mitochondria sort of break open and spill their contents, some of their contents into the cytoplasm of the cell. So we're watching this in real time under the microscope, and what's happening is that we're treating the cell with some kind of toxin that's going to stimulate this cell suicide program. In this case, it's 4.3% ethanol, and the cell undergoes the classic changes, visible changes that you can see. The cell shrinks, the nucleus condenses, the membrane is making all these little blebs that we showed schematically on those other slides. Now you can see them in a real cell as it's dying. And if we leave the cells in this ethanol-containing broth, they die. However, if you simply remove that broth and replace it with a nutrient broth that now does not contain the toxin, then what we can see, these are hours and minutes here, we can see that after 20 minutes, this cell looks a little bit better. It's, sort of coming back to its normal shape, and after an hour, it looks perfectly fine. Now, it's not just this one cell, or even one kind of cell. Here's an entire field of human lung cancer cells. As they're induced to undergo apoptosis, look, that one split into little pieces, and now we take away the toxin, and the cells recover their normal shape. They can go on to divide and survive for long periods of time. And you'll see here, if we watch this movie again, the cells are all dying. We change the nutrient broth, and they come back. And you can even see examples of cells dividing right before your eyes. Okay, so this is amazing. These cells can recover from this brink of death. What's even more remarkable and potentially very important is that they can actually survive even after their DNA has started to be damaged. Okay, so these are images. I won't tell you how this test really works, but it's a test for DNA damage. And we can see that untreated cells all look like these nice round circles, and that means they don't have DNA damage. The dying cells have these comet tails. It's called a comet assay. They appear to have these comet tails, and so that's a sign of DNA damage. And then when we allow the cells to recover, it looks like they repair their DNA. Well, that's not totally shocking. We know that cells have the capacity to repair DNA. We have to repair DNA, all the time, just because life itself can be kind of dangerous, and you're going to have, damage to your DNA from time to time, and so cells need the capacity to repair it. But it's really remarkable that they can repair that much damage. So, not every single mistake is repaired perfectly. We can see here that most of the cells are repairing most of the damage, but you can well imagine that occasionally a mistake slips through. And when that happens, you can have a variety of consequences, but one of them would be mutagenesis, right? So a mistake in the DNA that's not repaired would cause a mutation, a change in the DNA. And so we think that this ability of cells to recover from the brink of death is really a cell survival mechanism, but at a low frequency, it's also mutagenic and therefore potentially dangerous. Okay, so this slide shows you what normal chromosomes inside the nucleus of the cell look like. That's our normal DNA. In this case, it's mouse DNA, but this is what it would look like normally in these chromosomes. And we can see that in a small percentage, 1% to 2%, which is what's shown here, in different types of cells that we've tested, 1% to 2% of the cells will show some kind of chromosome abnormality. So here we see this end-to-end linkage of chromosomes that you don't see over here. So this weird guy right here. So that means that, for the most part, most of the cells are recovering perfectly well, and they're going back to their normal daily life. And 1% to 2% of the cells now have some DNA damage. Now, most of that DNA damage may not be all that important. Not every single nucleotide in your genome, not every single bit of DNA is equally important, so maybe some of those changes don't matter too much. But we know that a few of them do matter, and we know that a very small fraction of the cells that go through this process end up acquiring the characteristics of cancer cells. Okay, so we think that possibly this mechanism might, at a very low frequency, cause cancer. And of course, maybe most of the time it's helping us repair our tissues and renew our tissues and save our tissues from permanent damage. But in very rare cases and over the course of a very long life, when we might be exposed to lots of different toxic compounds at various times, we might end up getting cancer. But we don't imagine that cells evolve the capacity to do something in order to give anybody cancer. That doesn't make sense. Cells evolve the capacity to do something for a beneficial purpose. So what might the beneficial purpose be? Well, as I've alluded to, we think it might be to help repair tissues when they've been subjected to some kind of radiation or toxin or something like that. We wouldn't want, let's say that a nuclear explosion goes off and we're exposed to some massive radiation, but very temporarily. You wouldn't want every cell in your body to commit suicide all at the same time, right? So the cell, we think, has the capacity to recover in order to prevent the permanent tissue damage that you would have if too many cells died. Okay, so it would be particularly valuable if the cells in your body that are hardest to replace could do this. So neurons, cells that aren't dividing anymore, neurons in your brain, for example, you'd want to hold on to those if you could. You might even prefer that they be a teeny bit damaged, but still be there than not be there at all. Same with the cells in your heart, cardiac myocytes. It's very debatable whether they can be replaced at all, but they certainly are not easy to replace. And so this feature would be really quite beneficial if those kinds of cells could do it. So we tested whether heart muscle cells, cardiac myocytes could do this. We took them from a newborn rat, and then we're following, in this particular experiment, we're following 3 visible signs, whether the cell's shrinking or not, whether the mitochondria are changing, whether the nucleus is condensing, and we have one sort of fluorescent tag we can put on it that only binds to the cells that are dying, okay? And so we can take these heart muscle cells from the newborn rat, and if we are just growing them in nutrient broth, we don't detect these signs of cell death. But if we treat them, again, with ethanol, we see that virtually all the cells express or show all of these markers of cell death. But if we wash it away and we replace this toxic broth with fresh, not toxic broth, then we can see that the cells recover and they stop showing all these signs of death. We think that's really exciting, and there are other groups that have made observations that we think are consistent with the idea that this helps animals repair your heart tissue if a blood vessel gets blocked temporarily. Okay, so not work from my lab, but work from another lab where the people would tie off an artery in an experimental animal like a rabbit or a rat or a mouse, and they'd tie off the artery so blood doesn't go to the heart for like 5 minutes. And if you tie it off for 5 minutes and you look immediately after that, you can see these signs of apoptotic cell death. But if you tie that artery off and come back a day later, they have a way of marking those cells that were undergoing cell death with this particular fluorescent molecule, or in that case, it was a radioactively labeled molecule. They come back a day later and they see this marker inside of healthy heart cells. And their conclusion was that cells can reverse the apoptotic process, they can reverse the suicide program. So we think that's really exciting because that's a really important physiological context. We might have little transient blockages, frequently over the course of our life, but this doesn't kill our heart because maybe the cells can recover. Okay. So apoptosis is kind of a weird word. It was coined, or it was invented by the group I showed you in 1976, and it's coined from Greek roots that mean falling to death. So we had seen this remarkable capacity of the cells to recover from the brink of death. We'd seen it in lots of different types of cells in response to different types of toxins and stimuli. And so we decided to give it a name. And so we call it anastasis, which is the Greek word for rising to life. Okay, so this observation that these cells can do this raises all kinds of crazy questions, right? Hopefully you've got some questions. Where and when does this actually occur in animals? Is this just something we can see if we have the cells in the lab, or is this something cells can do really in our bodies? How does it work? How in the world do they do it? How do they pull that trick off? And then can we develop the tools that would allow us to enhance or suppress it? Because just imagine for a moment, sometimes you might want to enhance it, sometimes you might want to suppress it. Let's say you had a heart attack and your heart cells were, there are waves of cell death that happen after a heart attack, and maybe you would want to give someone a drug that would enhance the ability to recover from the brink of death and preserve that heart tissue, or stroke, same thing. But maybe if you're treating someone with cancer, you wouldn't want to actually, that maybe the cancer cells are really good at bouncing back. We've certainly saw it with those human lung cancer cells. And maybe in that case, what you would want to do is give someone a drug that would suppress this possibility, this capability. So we have a lot of questions here. I can't answer them all for you today, but I can tell you some things. Let's think about asking the question, where and when does this actually occur in animals? Or if it does at all, does it actually occur in animals? And think about the challenge that we have here, because a cell that has undergone anastasis looks just like a cell that hasn't. And so if we open the animal and look, we can't tell. We can't tell if the cell, what its history has been. So we decided to develop a way of permanently marking cells that have undergone this transient near-death experience. So how do we do that? Well, I can't give you the details. Maybe you don't even want them, or maybe you do, but you can ask me later. But here's the general idea. The general idea is we know that these cells have brief executioner caspase activity, right? This enzyme's going around cutting things up inside the cell, and that happens for a period of time. And we engineered a system. We engineered our animals, in this case, fruit flies, so that this would cause a permanent genetic change in that cell. If it lived, then it would express the green fluorescent protein, and it would glow in the dark. Okay, so what were we expecting from this experiment? I would say that we were expecting initially that the animals that we had engineered in this way would not express any green fluorescent protein normally, because cells aren't supposed to survive from the brink of death. And we would have to treat them with something. We'd have to stimulate cell death and do it in a way that was only a brief exposure to something toxic, ethanol or radiation or something, and some kind of stress. And then we would look and see if we could see this fluorescent protein start to appear. But the big surprise for us, and I'm not going to walk you through all of these examples here, but the big surprise for us was that without doing anything to these animals at all, we could see this green fluorescent protein being expressed in the normal tissues of the fly. So, so what does that mean? Well, what we believe this, so we know that apoptosis, the cell suicide program, is part of normal development. We know that, we've known that for a while. And so what this really shows, in work that was published just a couple of months ago, is that almost as common as apoptosis is that cells during normal life can recover from the brink of death. And that's how we can end up with all of these fluorescent cells. So does this ever happen in animals? Absolutely. And it's much more frequent than we would have imagined. And so that was one thing that we learned. But what about this stress-induced form of anastasis? What about where we hit the cells with some stress that causes them to undergo apoptosis? Can we see evidence of recovery from that kind of stress-induced apoptosis in a real animal? And so again, the answer is yes. So in this experiment, this is again, these are our cells in a fruit fly, and we're looking for them to glow green, and if we don't stress them, not so many cells glow green. And if we expose them temporarily to a heat stress, and so here I'm talking about a temperature that's basically our body temperature. And flies like to live at the same kind of temperatures that we like to live, kind of Santa Barbara temperatures, they sort of like that. And so if you expose them to the temperature inside of our bodies, that's a very stressful temperature for them. And we know that that can cause some of the cells to die. But if we expose them just temporarily to that kind of temperature, and then look, we see that many of the cells actually survive and recover from the brink of apoptotic cell death. Okay, so our studies in the fruit fly have shown us that a number of different things. We think we see a lot of normal anastasis during development of the fruit fly, and we can detect this stress-induced anastasis as well. Okay, so what in the world is going on inside of these cells that are recovering from the brink of death? How do they pull this off? We wanted to understand that, and so again, you have to ask yourself, how are we going to figure this out? So here are these cells again that are undergoing, apoptotic death and then recovering from the brink of death. And what we decided to do was to isolate the molecules, the inside of these cells, at each of the steps along the way. Okay? So we're actually characterizing the concentration of every single RNA inside of these cells as they undergo the death process and then as they recover from the brink of death. And we're asking what changes, what characterizes the death and what characterizes the recovery. And so I'll just sort of summarize those results for you by telling you that what we see when we look at these molecules and we sort of categorize them is we see that the cells that are undergoing cell death show evidence of a stress response. No surprise there. They're growth arrested, right? So if you're dying, you're not going to grow. But here was the big surprise for us. We actually saw that genes that are induced during the recovery period are induced, some of the genes are induced first during the death period. And we call this a poise for recovery. So think of it this way. The cells aren't just dying, but at the same time, they don't know conditions are bad, like it's not good. But they don't know if it's going to get better or it's going to get worse, right? So the cells are activating the death program, but at the same time, they're kind of activating a survival program alongside it until they give up the ghost, until it's really clear that it's just not going to get any better. So we see evidence for them poising for recovery. And then we also see that the recovery process itself looks like a two-step process, something we didn't really know or anticipate. Again, we see in the first few hours of recovery, we see continued stress response. We see growth and proliferation, activation of the genes that were shut down during this growth arrest period. And then in the later stages, 8 to 12 hours later, the program changes, and what the cells are trying to accomplish changes. And now we see them activating a cell migration program. And altogether, if you look at this all together, this looks a lot like a wound healing response, which makes a lot of sense if that's what the cells are doing. So these kinds of activities, proliferating, growing, and migrating, are adaptive if you're trying to heal a wound. That's exactly what you want to do. You want the cells to proliferate and make up for the cells that really did die. You want them to migrate to fill in the gaps in the tissue. And all of that is perfectly adaptive in a wound healing response. It's exactly what you don't want your cancer cells to do. You treat them with chemo or radiation therapy, you're trying to kill them off, and now the ones that bounce back are proliferating more. They're dividing more. They're migrating and spreading through the body, and those are exactly the things you don't want them to do. Okay, so as I said, we propose and are interested in testing whether anastasis might be a mechanism to preserve cells that are difficult to replace, cells like neurons and cardiac heart cells. So, if so, then treatments that enhance anastasis could be protective, neuroprotective, or cardioprotective. Does the capacity of cells to undergo anastasis change with age? In an aging organism, are cells less likely to undergo anastasis? Could this be a reason why our tissues don't regenerate as well as we age. And if that's true, then treatments that enhance anastasis could also be regenerative. But on the flip side of the coin, anastasis might represent a mechanism by which cells bounce back from chemotherapy. So we give chemotherapy, most of the tumor cells die, but all too frequently, some of the cells bounce back and there's a relapse in that cancer patient. Is anastasis a reason for some of the cells being able to escape? And if so, then treatments that inhibit anastasis could improve the effectiveness of the existing chemo and radiation therapies. So in the lab, we're taking a whole variety of approaches, a multi-pronged attack on the problem, so that we can figure out how it works normally and how we might intervene to either enhance or inhibit it. And this is exciting because really it has implications for this huge range of areas. It's really kind of launching a new field. We think it has implications for regenerative medicine, heart attacks and strokes, Alzheimer's and Parkinson's, cancer, normal development, and even potentially evolution. So I want to be sure to acknowledge in my last couple of minutes, acknowledge the people who are doing this work in my lab, who are an amazing team of people that are fun to work with every day. And I didn't just say that because they're sitting here in the audience, but it's really true. And so Gongping Sun is a postdoctoral fellow in the lab who has been spearheading this effort along with Austin Ding, who's a PhD student who moved here with me from Johns Hopkins. Hogan Tang was a postdoc in the lab who made the initial discovery that cells could recover from the brink of cell death. Yebdar and Kelvin are technicians in the lab. Jesse Wong is a master's student. And there are other people I don't have listed up here who are working on this project. We've been blessed with ample funding from a number of different places, including the NIH, the National Cancer Institute, and a private company. And I just want to show you our lab. This was what it used to look before we got here and look like, and this is what it looks like now. So we are excited to be here at UCSB, and I'd be happy to stop there and take your questions.